Okay, so in this lecture we are going to discuss how T cells develop. And so just as a general overview of what we're going to talk about, this goes back again to the bone marrow. And in the bone marrow you have your lymphoid progenitor. And then this lymphoid progenitor will become a T cell progenitor. And this T cell progenitor will leave the bone marrow and will travel to the thymus. And in the thymus, it is known as a double negative thymocyte. Now, what does double negative mean? Now remember, we have two types of T cells, a CD4 T cell and a CD8 T cell. So double negative means it is CD3 positive, CD4 negative, CD8 negative. So because it's both CD4, CD8 negative, it's called a double negative. T cells will always express CD3, so that will not change. So we have a double negative thymocyte, and our double negative thymocyte is going to rearrange its T cell receptor, and it's going to develop either into a gamma delta T cell or it's going to develop into a double positive thymocyte. And so just like the double negative, the double positive thymocyte is going to be CD3 plus, CD4 plus, and CD8 plus. Now this double positive thymocyte has to make a choice, and it's either going to become a CD8 positive, and it's a single positive thymocyte, or it's going to become a CD4 positive, and this is now a single positive thymocyte. So the double positives will express both CD4 and CD8, and the single positives will have to make a choice. Are they going to express CD8 or are they going to express CD4? Now the single positive CD8 T cell will then become a naive T cell, and then will become our cytotoxic T cell, our CTL. So remember, this is our CD8, our cytotoxic lymphocyte. CD4s have a bit more that can go on. Our CD4 single positive can become a natural T regulatory cell. Now, natural T regulatory simply just means that it was generated in the thymus. And the CD4 T cell or can become a naive T cell, and then it's going to become eventually a helper cell. It can become a Th1 helper cell. It may become a Th2 cell. It can become a Th17 cell, or what we call an inducible T regulatory. And this occurs in the periphery. So we have our periphery over here, and this is occurring after the T cells become activated, and then everything else occurred either in the primary lymphoid tissues. We have our bone marrow on this end here, and then most of the T cell development, of course, will take place in the thymus. There's our bone marrow. Okay, so let's talk a little bit about the thymus. The thymus is going to be made up of three general zones. We have our subcapsular zone. We have our cortex. And you have the medulla. Now, each of these zones are going to be very important in T cell development, and the T cells are going to develop in this direction. There's a little arrow. So our T cells will develop in this direction. And so they're basically going to move from the subcapsular zone through the cortex and then into the medulla, and then eventually into the circulation if they are released. There are some cells that are very important to point out, and we will continue to talk about these cells. First off, these little blue balls are our thymocytes. And these cells are going to be proliferating when they arrive. So what you can think about in this respect is over here we have our double negative thymocytes, and they're going to proliferate. And your body does this because it wants to make a lot of different clones of T cells, because the actual T cells that actually leave the thymus is very low because they have to get through a selection process. So in your bone marrow, you make the thymocytes. They come to the thymus and they proliferate into more double negative thymocytes. Now, the double negatives haven't yet rearranged their T cell receptors, so they're not specific for a particular antigen. You're basically just increasing the potential of having a successful T cell develop if you make more clones at the stage prior to T cell receptor rearrangement. And then you're going to have in the middle, in the cortex zone, you're going to have these green cells, and these green cells are known as cortical epithelial cells. Now, these cortical epithelial cells are going to be important in a process we'll talk about. It's called positive selection. So in the cortex is where positive selection occurs. And in the medulla, you have many cells that are important. You will have in the medulla, you'll have your traditional dendritic cells. You will have your traditional macrophages. And you'll have your medullary epithelial cells. And all of these will be in the process of negative selection. So in the medulla is where you have negative selection. Now each of these has its own role in negative selection. So our dendritic cell's role is going to be present antigen. Our macrophage's role is going to be kill self-reactive T cells. And our medullary epithelial cells, their role is to generate we'll say extra thymic antigens. And so what I mean by that is that your cells in your medulla are able to express um, proteins that are not part of the thymus. So a good example is insulin. You don't want to release a T cell that's specific for insulin, and so in the thymus, your medullary epithelial cells will express insulin so that in negative selection, your dendritic cells can present insulin, and any T cell that binds to that antigen will be eliminated before they can get out into your periphery, where, of course, insulin is very abundant. And so your medullary epithelial cells, you can think of them as making a snapshot of your body. And this is important for T cells because keep in mind that B cells go through negative selection as well, but it's not as rigorous as T cells. And that's because there's another layer of control for B cell activation, and it's called T cell help. So if you potentially have an autoreactive B cell that gets loose, as long as it's not able to see help, it's not going to do much auto damage. If, however, you release a potential auto T cell, then you could potentially have a very debilitating autoimmune disease. And so your medulla is the process of negative selection, picking out clones that respond to self-antigens and deleting them before they get into the periphery. Okay, so those are the main areas of the thymus, and those are the main cells of the thymus. So now let's talk about the development in the thymus of your T cells. So you're going to have a series of steps in order for your T cells to become a mature T cell. Now there's, in general, two types of T cell. So we have our alpha beta T cell, which is approximately 90 to 95% or more. And then we have our gamma delta T cells, approximately less than 10%. And so both of these are going to develop initially in the thymus. So let's talk a little bit about how this works. So again, we were in the bone marrow, and then we came to the thymus, and now we are a double negative thymocyte. And in this double negative stage, we're going to do our beta, alpha, and delta rearrangements. And so we can become a gamma delta T cell. It's a delta. We also have to add gamma. And in this case, this T cell will leave. The gamma delta T cell does no positive selection and no negative selection. Now this double positive or double negative thymocyte will then move onto the next stage. It's supposed to be gamma, beta, gamma, delta, not alpha. Sorry. And it will move to the next stage of the double negative stage, and this is to do alpha rearrangement. And then once it does the alpha rearrangement, it becomes a double positive thymocyte. And it's going to express CD3, CD8, and CD4. And then it's going to go through positive selection. And it's going to become a single positive thymocyte. And then it's going to undergo negative selection And it's still going to be a single positive thymocyte that gets released or it gets killed. Now, just as I'm going to circle things here. Let's see. We're going to have the subcapsular are going to be the double negative thymocytes, the double negative rearrangement, and then the cortex is going to be where you have your positive selection, and then the medulla is where you're going to have your negative selection. And so that's sort of how the progression of T cell development occurs in the thymus. So let's just talk very briefly about some molecules that are important on the surface. You can see right away that we have our CD3 molecule, and our CD3 is always going to be expressed. That never changes. Similar to our B cell receptor, when you're rearranging your VDJ rearrangements or VJ rearrangements on your beta, delta, gamma, or alpha chains, you're going to have RAG activity, the recombination activating gene, and the deoxynucleotidal transferase activity. But once you have a pre-T cell, or once you have a double positive T cell, once you have a alpha beta chain on the surface or the gamma delta on the surface, those genes get turned off and are never turned on again. You have beta, gamma, delta genes that are going to turn on first. Beta gene will go to the surface and be at the surface for the remainder of the life of that T cell. The light chain rearrangement occurs after you get a successful beta chain rearrangement. And after you get a successful light chain, you get expression on the surface for the life of that T cell. During the double positive, you have both CD4 and CD8. On the single positive, they have to make a choice. Are they going to be CD4 or are they going to be CD8? So let's just talk a little bit more about T cell rearrangements. So there are two chromosomes that are important for our T cells. We have chromosome 7, which encodes T cell receptor beta, and it also has the T cell receptor gamma region. And then we have T cell receptor delta, which is the green down in the bottom, and we have T cell receptor alpha, which is the red. And so basically what's going to happen, the steps are going to be, 1, you have the beta competes with the delta plus gamma rearrangement. And if this is successful, you go to 2 alpha rearrangement. And this success is about 90 to 95%. If this is successful, you get a gamma delta T cell. And this will travel to mucosal tissues. And the alpha rearrangement will undergo double positive, to positive selection, to a single positive, to negative selection. And then it will be released in the periphery. So again, this is just showing that the chromosome 7 has both the beta and the gamma rearranging, and so they're going to compete and rearrange at the same time. Chromosome 14 has the delta and the alpha chains, and there is overlap between the delta and alpha chains. So if the beta wins, the delta basically regions get clipped out of chromosome 14. If the beta wins, the gamma regions get clipped out of chromosome 7. So that particular T cell could never go back to become a gamma delta T cell. It's always going to be an alpha beta T cell. The same thing if the gamma delta wins, the beta chain and the alpha chains get clipped out of those chromosomes, so you can only have one type of T cell. Okay, so let's just talk about positive and negative selection. Now, when you think about this, you want to think about it in terms of the actual T cell. So, positive selection. This means a T cell is selected for its MHC1 or MHC2 recognition. Okay, and so what's going on here in positive selection, and this occurs in the cortex with the cortical epithelial cells. So basically, we'll have a cortical epithelial cell. This is not an APC, okay? So no co-stimulation. So basically, in essence, what this means is the cortical epithelial cell cannot activate these T cells. All it can do, like any other nucleated cell, is express MHC on its surface. Now, the cortical epithelial cell is unique because it can express both MHC1 and MHC2. And so it'll have on its surface, for example, an MHC2 molecule, and it'll also have on its surface an MHC1 molecule, beta microglobulin. Oops. Okay, so our single positive thymocyte that decides to interact with your MHC2, or I'm sorry, double positive. So this is a double positive thymocyte, and it has its T cell receptor. And it interacts with MHC2, is going to develop into a CD4 molecule. So this one will leave and become a CD4 single positive. Now our T cell that decides to interact with our MHC class 1, and it's a double positive, and this is its T cell receptor. It's going to interact, and CD8 forms dimers. I'm not going to draw it that way. So this is going to be CD8. And so this will lead to a CD8 single positive. So you have a double positive CD4, CD8, and that will interact with either a MHC class 1 or MHC class 2 on the surface of this cortical epithelial cell. Whichever molecule this double positive sees first, it's going to become a single positive. Now, the cortical epithelial cell, again, is not a traditional antigen-presenting cell, so it's not activating them. All it's saying is, can you recognize self-MHC? So it's really looking for self-MHC recognition. And the reason for this is because if you're going to release T cells into the periphery to prevent infections, the only way they can do this is if they see peptide plus the MHC molecule. So positive selection makes sure that they can see the MHC molecule. It doesn't have anything to do with the peptide. It's only with the MHC molecule. And so now we have our single positives. And so they're going to move into the medulla. So over here we have negative selection. And negative selection is deleting self-reactive clones. So now that they can respond to MHC, self-MHC, now your body has to make sure, well, that's great that you can see MHC molecules, but now we don't want you to attack yourself. And so you're going to have a traditional antigen-presenting cell, our dendritic cell. So here's our DC. And the dendritic cell is going to express on its surface, again, this time it's a professional antigen-presenting cell. So it's going to have on its surface MHC class 1. And on MHC class 1, it's going to interact with the T cell receptor for CD8. And you're going to get the CD8 molecule. Now, this time, you are looking for peptide. So here's the peptide. Now, if the interaction is weak, the T cell survives. If the interaction is strong, the T cell is deleted. And that's because if the T cell recognizes MHC but not the peptide, it's going to bind for a small period of time, but then it's going to move on and get released. If, however, the CD8 T cell receptor binds to the antigen very tightly in the thymus, it means that that T cell is specific for the antigen. And that T cell will get deleted. And this will happen with the CD4 T cells as well. So for CD4 T cells, there's also MHC class 2. And you have the same story. Strong T cell, MHC class 2 with antigen interaction gets deleted. Weak gets released into the periphery. Okay, so now let's move on into activation of actual T cells. So in the periphery, the T cell will become activated, and you're going to get an immunological synapse. So you have on one side, you have our, make it blue again, our T cell. And the T cell is going to have a T cell receptor. And the T cell also always will express CD28. And then, so your antigen presenting cell, which is much larger, your antigen presenting cell is going to have on its surface the MHC molecule. And it's going to have this antigen. That's going to fit into the T cell receptor. Your T cell will have CD4. This is a CD4 cell. So we'll have CD4 interact with your MHC. And so we have here signal 1. Now signal 2 is going to be caused by color, purple, by the expression of CD80, which is also CD81, can be called that. And so this is going to lead to signal 2. So interaction with the T cell receptor with its antigen is signal 1, interaction with the co-stimulatory molecule CD28, CD80 is signal 2. Now the other thing we're going to have is tolerance. Oh, sorry, I had it here. Okay, so we have our signal 1, we have our signal 2, we understand that. It's supposed to get small. So we'll put that guy over there. Now for tolerance, tolerance is going to be at many levels. The first is going to be signal 1 without signal 2, and you get anergy. And anergy is the same thing. It's going to upregulate FAS, and you're going to get apoptosis. Because that's potentially a self-reactive clone. Remember, when you're not infected, your MHC molecules are going to be loaded with self-peptides. So if you have a T cell that recognizes self-peptide and there's no infection, so there's no beta-7 or CD80, I should also say it's also called beta-7. There's no beta-7 or CD80 signaling. You don't get that signal 2. That T cell needs to be deleted, and it gets deleted through apoptosis by becoming anergic. Now, something else can happen. You can get signal 2 without signal 1. And in this case, nothing happens. And in this case, it's because, for example, you have the flu, and so your antigen-presenting cells are expressing the CD80. They are expressing flu antigens, but you have a measles T cell that comes along, and it sees the CD80 because it has CD28 on its surface, but it doesn't recognize the antigen, so it simply just goes away. There is no anergy in that sense because you don't want to delete clones that you might need for another infection because you have a different infection going on. So in this case, there is no reaction. The T cell just has to keep roaming around looking for its antigen. Now there's also Tregs. And so we have our natural Tregs. Oops. And these came from the thymus. And then we have our inducible, or our iTregs. And this is in the periphery. Now, one thing to keep in mind about tolerance is we have central tolerance, and we have peripheral tolerance. Now, central tolerance is going to be dependent on a gene called AIR. It's the autoimmune, it's basically that autoimmune gene that is able to turn on and make that snapshot of your body inside of your thymus. In your peripheral tolerance, you're going to rely on T regulatory cells, and T regulatory cells express FOXP3 and CD25. And what's interesting is central tolerance does delete delete self-reactant T cells. However, peripheral tolerance is actually extremely important. Usually people who have deficiencies in their Tregs are not able to survive beyond a year of life. Your inducible Tregs, they're basically when you have a potential self-antigen that they recognize, they can become tolerant to that antigen and become a Treg, so that if another T cell comes along and is specific for that antigen, they can actually suppress that T cell's response. So peripheral tolerance is at that level as well. Now another peripheral tolerance, let me move this up, is going to be at the level of immune privileged sites. And so this basically means there are some areas of your body that your immune system just does not go to. So your immune system will not go into your brain, your eye, your testes or ovaries. And it's because the body does not want your immune system to have inflammation at these sites because that causes a lot of damage. However, if you do have an infection in your brain, that you will get your immune system in there. The eye as well. If you have inflammation of the eye, it's very important to treat because typically once your immune system gets into one eye to try to clear an infection, you can go blind, and then the immune system can now enter the other eye, and you can go blind in both eyes. There's also overstimulation. So against too much antigen binding will cause also clonal anergy as well. Now, finally, I want to talk just a little bit about T cell subtypes. So we have, again, we have our CD4 helpers. And these are going to be Th1, and this is primarily for intracellular. We have our Th2, which is going to be for our extracellular parasites, worms. It's doing that. We have our Th17, extracellular bacteria, and we have our Tregs, which are our natural Tregs or our inducible Tregs. And then for our CD8s, these are cytotoxic. So they're going to kill specific targets. So this is MHC1 plus peptide. And they can also kill FAS expressing cells, similar to like a NK cell. And then finally we have our gamma deltas. Just a quick word, these are going to be minor. We don't really cover these too much in these first immunology courses. They develop in the thymus. They do not undergo positive or negative selection. They are innate-like. Because there's no positive selection, they don't need to interact with MHC. They are considered a first line of defense in the skin and gut mucosa. They recognize common microbial pathogens. So they're similar to innate. They recognize sort of just common antigens. Okay, and that's all. Thank you.